Alright, so this video is a continuation of section 2 of chapter 5 in biology. We're working on this second objective. We're trying to follow chromosomes through the process of mitosis and cytokinesis. So we'd gotten through a lot of things already, but we got to this slide here where we were talking about mitosis, and we even got through a picture of prophase already, but I'm going to go through that again just to make sure that you have everything that you need. So prophase. Our key word for prophase is going to be to prepare. Before you enter mitosis, you're in interphase. Do that again. And in an interphase, you have a nucleus, but you can't see the DNA. One of the first things that happens from interphase entering prophase is that DNA condenses so that you can see it. So I have those characteristic X's for my chromosomes in here. I can see my DNA where I couldn't see it before. So if I go back a slide here, that DNA, the chromatin, is going to condense so that now I have those chromatids that I can see. Other things that are going to happen in prophase, that nuclear envelope is going to need to break down. You're also going to have centrioles that are going to migrate to opposite ends of the cell, and you should also see some spindle fibers that we're going to use later. So I need to add a few things to my picture here. A couple of centrioles, some spindle fibers, and then I need to erase parts of my membrane, that nuclear membrane, because it's starting to break down. So that picture is for prophase. Again, my key word for prophase is to prepare. I'm trying to prepare for that nuclear division. After prophase, you have metaphase. And metaphase is going to have a key word here, middle. When I say middle, I'm trying to help you identify this stage because it's the easiest one to identify. During mitosis, in metaphase, your chromosomes are going to line up in the middle of the cell. So if I have this equator here, the spindle fibers that attach to the centromeres of these chromosomes are going to push and pull on those chromosomes until that you end up with all three of these chromosomes right now that I've drawn lined up in the middle. So your picture ends up looking like this. It's pretty easy to identify. But again, the purpose for this aligning those chromosomes in the middle of the cell is basically just to make sure that your chromosomes don't get all tangled up when you're trying to pull them to opposite ends of the cell. So if the next step, realistically, I'm trying to separate these two chromatids here, these sister chromatids, and then pull one way while the other moves the other way to opposite ends of the cell, I can't have those running into other chromosomes along the way, or else they're going to get tangled up, or else there could be some damage that results from that. So I line up all the chromosomes in the middle so that I can avoid that problem. Right? So that would be metaphase, keyword middle. You're just looking for chromosomes lined up in the middle of the cell. After metaphase, we have anaphase. Anaphase is when you're going to take those sister chromatids and separate them, pull them apart to opposite ends of the cell. So my keyword here is apart. My spindle fibers are going to shorten and you're going to pull those sister chromatids away from each other towards the opposite ends of the cell. So in the picture that we're drawing, I need to see a cell. Those sister chromatids have divided, and they're getting pulled towards the opposite ends of the cell. One of the things that you're going to see in anaphase is a characteristic V shape as those chromosomes move through the cytoplasm. They're being pulled by the centromere, by that central point, but those tails, those ends, drag behind while you're pulling them through the cytoplasm. So you should always see those characteristic V shapes in this phase, because that indicates that those chromosomes are moving. They're actually getting pulled away from the center. So that is anaphase. Your keyword is apart. That's what your picture needs to look like. The last phase of mitosis is telophase. Telophase is also going to have a keyword for us. This time it's going to be 2, the number 2. Right? Once you have a complete set of chromosomes moved to each end of the cell, you're going to start forming new nuclei to surround those chromosomes. So our picture is going to look a little bit like this. That's the next part of my cycle here. I'm actually going to draw a little bit of furrowing in this picture. That actually gives us a head start on the cytokinesis, the next step. But I've pulled the chromosomes to opposite ends of the cell, so I have some chromosomes over here, 3 of them still. I'm going to start forming my nuclear membrane around those chromosomes. But then on the other side, I'm going to draw it like I'm a little farther ahead. Maybe that side, that nucleus is complete. I've finished building that nuclear envelope. And I'm not going to draw DNA, because that last thing that's going to happen in telophase, that DNA is going to start to decondense, go from chromatid to chromatin form. So I shouldn't really be able to see it. I shouldn't really be able to see that DNA at that point. So going from top to bottom here, that first nucleus is just starting to form. I can still see that DNA. The other nucleus has already finished that process of forming, and my DNA has started to decondense. It should be invisible at that point. So this is my picture for telophase. Went the wrong way. And telophase is where you have 2 nuclei again. So if you see 2 different nucleus inside of that cell, 2 different nuclei inside of that cell, you know you're in telophase. Again, that last step, your chromatids are going to uncoil so that you're left with that chromatin form where you can't see the DNA anymore. All right. So that's my 4 phases of mitosis, but I also need to complete that division by doing a cytokinesis. So what happens in that last step, I'm going to take this cell that's formed, or these 2 nuclei, this cell here, this furrowing that I drew to begin with, you're essentially going to have the membrane joining up from one side to the other. And the easiest way to describe that is just like you had a drawstring around that cell. So just like a drawstring in a hoodie, when you pull it tight, it closes up. So you have this protein band around that cell that's tightening up, and that helps you pinch those 2 sides of the membrane together. And ultimately, right, in cytokinesis here, once that's pinched close, those membranes are going to fuse, and that's when you have 2 distinct cells. So cytokinesis is technically the last step of mitosis. You have to pinch off that membrane to form 2 new cells, 2 distinct cells. So in this picture here, I've kind of combined telophase and cytokinesis into one. But remember, only after cytokinesis has occurred do you really end up with those 2 new cells both of which are going to be an interphase all over again. Remember, this one over here is our interphase. It'll stay in interphase for a long period of time, but eventually that cell is going to be big enough again that it needs to divide again. So you'll enter mitosis and go through the whole process again. Prophase, metaphase, anaphase, telophase. But you need these 4 pictures in your notes so that you can identify those different phases later. The only thing left to take care of is to understand that plant cells are different than animal cells. So this last thing that we need to fill in here, remember, plants actually have a cell wall. So I'm going to draw another picture here. So that box shape right there is my cell wall for this plant cell. It also has a membrane surrounding the cell. But at the end of telophase, I'll have 2 nuclei on one end and the other, and I need to separate those 2 cells, but I can't do that same drawstring action I did before. The cell wall is rigid, so I can't pinch that off to form 2 new cells. I actually have to build a new cell plate right up and down the middle of that cell. So I'm going to build this cell plate that eventually turns into a cell wall, separating those 2 plant cells in the long run. So plant cells, again, they're a little bit different than animal cells. The other major difference is that plant cells don't need those centrioles. Right? I had centrioles in my animal cell over here because I needed an anchor point to pull from. But I don't really need that in a plant because plants can just pull anchor from that cell wall itself, so they don't really need that to make this whole process work. Otherwise, plants do the exact same things that animal cells do. They still have prophase, metaphase, anaphase, telophase that you can identify. It's just their cytokinesis is a little bit different. They're going to build that cell plate. Right up and down the middle of the cell. Eventually, that new cell wall forms, and you have 2 distinct cells that are separate from each other. All right, so that's all we had to finish up.